Today I'd like to talk about Cu62, the Polycystin-1 interacting protein 1 protein. Polycystin-1 is a product of the PKD1 gene, which is mutated in 85% of cases of autosomal dominant polycystic kidney disease. Polycystin-1 is a large protein that threads through the membrane 11 times and has a large ectodomain that's mainly composed of PKD repeats. Cu62 interacts with these repeats. Cu62 was discovered when we compared the exosome proteome of individuals that had proven PKD1 mutations with wild-type individuals. This study showed that the levels of polycystin-1, polycystin-2 and Cu62 were decreased in individuals that harboured PKD mutations versus wild-type. Cu62 has a signal peptide cleavage site, but lacks a transmembrane domain and has 5 end-linked carbohydrate attachment sites. And this can confer 18 kilodaltons of carbohydrate when the protein is expressed in 293 T cells. It can interact with itself, and it can interact with PKD domains. And here we look at the trimer of 15, 16 and 17 versus Cu62. And what we see here is that on IP there's a high molecular weight complex of the two proteins which is resistant to SDS and high temperature, 65 degrees. So we made two monoclonals to Cu62, and one of the first questions that we asked was, is it present on primary cilia? Polycystin-1 and polycystin-2 have been localized to primary cilia, and Cu62 seems to interact with them on the primary cilium. But it's not the only place you find Cu62 or polycystin-1. In migrating cells, PC1 perfectly co-localizes with tetraspanin 4. Tetraspanin 4 is a marker for the retraction fiber and the migrosome. Migrosomes are specialized extracellular vesicles that are involved in removing senescent or defective mitochondria from migrating cells. These cells are also megalin positive, implying that they're proximal tubule cells. We also think that polycystin-1, fibrocystin, and polycystin-2 and Cu62 are also present on focal adhesions with alpha-actinin and talin. And returning to the retraction fiber, the retraction fibers are mainly polycystin positive, but the migrosomes are intensely positive for Cu62, which forms a shell around the polycystin-1 on the migrosomes. So there's a profound defect in mitochondria in cyst-lining epithelial cells, and we wondered if treatment of cells with a mitochondrial toxin would alter the distribution of polycystin-1. When we give CCCP, which uncouples the proton gradient in the mitochondria begin to disintegrate. And around some of the disintegrating mitochondria, we see these nidi of polycystin-1 with a core of Cu62. And at the edge of the cell, we see Cu62 and mitochondria. Probably these mitochondria are being extruded as migrosomes. If we allow these structures to develop, they form rings, and we call these fairy rings. And embedded in them are the mitochondria, and there's a core of Cu62 in the middle. So that's migrating cells. In fully polarized cells, we see these fairy rings, and we also see these large extracellular vesicles being budded apically. And on close inspection, there's a reticular network of polycystin-1, Cu62, and polycystin-2 in the actual membrane of these vesicles. So our running hypothesis is that the mitochondrial defect seen in cyst-lining epithelia may be due to a defect in the polycystin complex in that it cannot identify, sequester, and then extrude defective mitochondria, so that they build up within the cell, giving rise to the various biochemical and morphological features seen in the mitochondria of cyst-lining epithelia.